sure. I think it's this one. Okay, yeah.
Okay, it's a pleasure for me to introduce our today's speaker, Anacs Bушниta from the University of Texas in El-Paso. And Anas will talk us about mathematical modeling of multiple myeloma. Please, Anas, you can begin your presentation.
Thank you very much, Vitaly, for the introduction. And thank you also, Anastasia, for helping me with all the logistic operations and preparation for this talk. So before starting, I would like to give credit to all these the following colleagues. So all of what I'm going to present is based on joint works with these colleagues, including Professor Vitaly Volpert. So regarding the outline of this talk, so I will start by talking about the biology or biological background of multiple myeloma. Then I will introduce two different methods for modeling multiple myeloma intraclonal heterogeneity, which are multi-scale modeling and phenotype structured modeling. Then I will talk about another application of multi-scale modeling where we use different simulations to determine treatment strategies that overcome multiple myeloma drug resistance. After that, I will talk about some modeling works that we did for the infiltration of multiple myeloma and its impact on erythropoiesis and anemia. And I will conclude by talking about some general conclusions and also perspectives for future work. So multiple myeloma is a type of hematological cancer. So it's characterized by the excessive proliferation of malignant plasma cells. So these are major B cells that infiltrate to the bone marrow and start disrupting what's going on there. Basically, they disrupt the process of hematopoiesis, which is the production of blood cells, but also bone remodeling. So bone remodeling is the process by which the bone regenerates, so through resorption of the bone and production of a new bone. So each year we have around 140,000 cases of multiple myeloma, and most of these cases are reported in the developed countries. And the symptoms of this disease include anemia due to the lack of red blood cell production, bone pain because of disruption of bone remodeling, renal failure because of increased calcium, weariness, hypercalcemia, infections, weight loss, and other. So here we can see some, I think, MRI or CT scan where we can see that these patients had a lesion of multiple myeloma in this region, and also they had a bone fracture. Okay, so one particular aspect of multiple myeloma is that it evolves very fast. And basically, multiple myeloma is the final stage of several diseases or several clinical cases. So first, we have a clinical case called smoldering, we have monoclonal gammopathy of uncertain significance, MGUS. And then we have smoldering multiple myeloma, and then finally, we have myeloma. And when we analyze the multiple myeloma tumors, we find that they consist basically of several clones, typically between 3 and 7 clones, and sometimes more than 7 clones. And when we observe the evolutionary dynamics of these tumors, we observe linear evolution and branching evolution patterns. So basically, for linear evolution, it means that we have cells that are that acquire mutations, like mutation after mutation, in a sequential manner. And in the branching evolutionary pattern, that we have different lineages of cells that harbor different mutations and that evolve in parallel. And sometimes we have both of these dynamics in the same tumor. So when we look at the genetic landscape of multiple myeloma, we find basically two main translocations. So translocations is a set of mutations that represents the triggering events for the development of multiple myeloma. So there is this translocation T414, which is the most proliferative one, and there is a less proliferative translocation called T1114. And basically, there are mutations, different mutations. So as you can see here in this graph, so this is like the intracellular regulation of myeloma cell. And there are different proteins and pathways which are altered due to mutations, and in particular, the RAS, RAF, ERK pathway, where we see the existence of the NRAS, KRAS, and RAF in around 20% of cases. And then we have also some other mutations, such as the ones in the AKT-PI3K pathway, P53 MM sets, and IRF4 proteins, and many others. So what are the treatment options for multiple myeloma? So basically, the treatment of multiple myeloma is not successful in most of the times, unfortunately, and this is due to the intraclonal heterogeneity of multiple myeloma. So basically, when we use chemotherapy, then we cannot eliminate all the cells, because there are some cells which belong to clones which are more resistant to the treatment, and this leads to relapse. So the strategy for the treatment consists of two phases. So the first one is induction, where we have a short-term and intensive treatment of basically 3 drugs that induce apoptosis. So these drugs are proteasome inhibitors, immunomodulatory drugs, and corticosteroids. And after that, we start administrating consolidation and maintenance treatment. So in consolidation treatments are also intensive, and maintenance treatments, they are low dose, but with different treatments. And the objective is to prevent relapses. So the objective is to keep the number of cells low and to prevent the development of high symptoms, and to basically increase the life span and prevent the evolution of the disease. So there are some other options for the treatments which are currently under development and under testing clinical trials, basically radiotherapy, immunotherapy, and targeted chemotherapy. So for targeted chemotherapy, so sometimes some patients, they do not have some mutations, and they can be responsive to some type of treatments, such as, for example, the tyrosine kinase inhibitors, which I will talk about in this talk. But these are the patients that do not have a mutation in the RAS or RAF proteins. Okay, so let's start with the modeling of intraclonal heterogeneity. So the objective of this work was to develop mathematical model that describe this experiment. So in this experiment, there is an evolution of multiple myeloma tumors, which was documented through whole exome sequencing and single cell genetic analysis. So we could see that there are, so as you can see here, there are this first clone, and then there is the second clone with a new mutation. So this second clone has 2 mutations, ABCA and FAT. And then there is this branching evolutionary pattern. So there are some cells that will acquire an NRAS mutation, while there are others that will acquire a KRAS mutation. And these ones will also acquire further mutations, in particular the one with IRF4. And so there are like 2 lineages of multiple myeloma cells that coexist. One lineage has an NRAS mutation, and the other has a KRAS mutation. So we try to reproduce these experiments by developing a specific multiscale model. So the multiscale model include a bone marrow stromal cell. So this is a screenshot of a simulation. And basically this big cell in the middle is what we call a bone marrow stromal cell. So these are like cells in the bone marrow that secretes growth factors, which promotes the proliferation and survival of multiple myeloma cells. And then we have all these different multiple myeloma cells that surround the bone marrow stromal cell. And as you can see, they are represented by different colors. So each color represents a specific clone. So the blue cells belong to clone 1. The yellow cells belong to clone 1. The blue cells belong to clone 3, I think. And the purple cells belong to clone 2, and the cyan cells belong to clone 4. So first of all, we in this model, we have cells represented as individual objects. So basically, they are modeled as soft spheres, and their motion is tracked using Newton's second law of dynamics. So basically, we suppose that each cell is subject to two types of forces. So the first one is cell-cell interaction. So basically, the cells, they push each other. And also we have a chemotactic force, which basically allow the cells to get closer to the bone marrow stromal cells. So the bone marrow, the multiple myeloma cells, they can sense the gradients of growth factors, and they can move toward the bone marrow stromal cells to increase their chance of proliferation and survival. So how do we model these extracellular proteins secreted by bone marrow stromal cells? We do this using this specific partial differential equations. So these are proteins secreted by bone marrow stromal cells. And as you can see, we have a production term, we have a consumption term, and we have a decay term. So here we model two types of growth factors. The first one is basically used by multiple myeloma cells for their chemotactic movement. So this is the SDF1, this one. And the IDF1 is the growth factor which promotes the survival, or I think it should be IGF. So this is a typo, IGF1. It's the growth factor which promotes the survival and proliferation of multiple myeloma cells. And inside of each cell, we have a system of ordinary differential equation, which represents the intracellular regulation of cells. So this is a multi-scale model, which means that we have some, at the same time, we track the concentration of proteins outside of cells, and at the same time, we also describe what's going on inside of cells. So here we have these three equations, and they describe the regulation of the MIPK or RAS ERK and AKT-PI3K pathways. So these are two pathways which are involved in the proliferation and apoptosis of multiple myeloma cells. So as you can see here in this graph, and basically we have this RAS ERK pathway that leads to proliferation, and then we have this other cell, this other pathway which is responsible for apoptosis. And we introduce the effect of the mutations by considering that the rate of ERK activation depends on a function that I will explain next. So this is the main innovation in the model, which is that we track the distribution of the phenotype of cells by using a specific equation. So we have a specific equation that links the genotype of cells to their phenotype, which is here, which here corresponds to the activation of the ERK protein, and this is due to the higher survival of the RAS protein. So basically, this is what this function looks like, or the function for the RAS activation rates. And as you can see here, we have each of these clone is represented by a specific area where the activation rate is sufficiently higher. So this represents the fitness landscape. And this graph shows the different clones that we are considering. So there is this first clone, and then there is this branching pattern. So we have this clone with the NRAS, and then we have these two clones with KRAS, with this fourth clone having an additional IRF4 mutation. So basically, we just reproduce what's in the experiments. So these are some screenshots of simulations, and basically, during the first hours or the first days of the simulation, what happens is that the cells start going or migrating toward the bone marrow stromal cell, and this is the effects of chemotaxis. And here, something which is interesting is that some cells will not reach the BMSC and will die because they will not have enough resources, while others will come close to the bone marrow stromal cell and will start differentiating. And because of the effects of mutations, so here I forgot how we introduce mutations. So basically, mutation is introduced by a random perturbation that we added to this function after each division. So when a cell divides, then the daughter cells will inherit the value of this variable, the genome Z, plus a perturbation, a random perturbation, which corresponds to the epigenetic mutation. So as you can see here, we can see the apparition of a few clones, so represented by different colors. Mainly, they belong to, I think, clones number 2 and clones number 3. Okay, so what happened when we run the simulation for a sufficiently long time is that we start seeing the different phases where there are different composition of the clones. So basically, when there is a new clone that starts appearing, if this clone is sufficiently aggressive, then first of all, the total size of the tumor will increase. So the total size, the tumor will expand, and then this the composition, the clonal composition of the tumor will change. So as we can see here at the beginning, there was mainly clones, the clones numbers 3 and 2, but toward the end, it's the clones number 4, which is here represented with this magenta color, and the clone number 2, which is represented by the cyan color, which are represented. And also we have some cells that do not belong to any clone, which are represented in the blue color. So what happened when we look at the composition of or the population dynamics of different clones inside of the tumor? So we see that first of all, there is the emergence of clone number 2, which is very aggressive. So this clone quickly rise to predominance. And after that, the clone number 3 appears, and it paves the way to the emergence of the clone number 4. And when clone number 4 emerges, then we have these two clones basically that consist most of the cells belonging to the tumor. And basically we can see that after the emergence of each clone, there is an increase in the size of the tumor. So basically, when the tumor consisted only of clones of the clone 1, then the number of cells was approximately between 400 and 500. And then after the emergence of the second clone, then the size increases. And finally, so after the emergence of each clone, the number of cells in the tumor increases. So these were the main results of this study. And in general, I think the main effects that was considered here is the competition for limited resources. And we can show that the most aggressive clones will basically compete for these limited resources and will be able to consist most of the composition of the tumor. And also it is important to note that this model includes few sources of stochastic noises, such as the one related to the cell cycle and the mutations and so on. And this is important because when we develop the continuous model, we will see that these stochastic noises can have an important effect. And basically, these predictions were consistent with the experiment. So we did some comparisons for the composition of the tumor at different stages, and they were quite consistent. Okay, so that was the first model, and the second model used a different approach, which is phenotype structured modeling. So for phenotype structured modeling, it's a continuous model. So basically we try to represent the population of cells, but the novelty of this model is that we also show how this population is distributed across the genome. So it's like partial differential equations, but we don't, instead of considering the space, the actual space, we are in fact considering the genotypic space. And basically, this method was developed for the other applications, in particular in ecology by Professor Vitali Volper to show the emergence of species, but also it was also developed by other research groups and applied in other contexts, even in cancer. So here we are trying to develop a model that uses this approach and try to consider the effects of natural selection, which is mediated by different effects, such as the limited concentration of growth factors, the effects of the treatment, and natural death, and see how this selective pressure changes the composition of the tumor or the clonal composition of the tumor. So this is what the concentration of cell looks like. So basically we have two terms. The first one is a diffusion term, and the second is basically a function which stands for natural selection. So the diffusion term basically corresponds to the effects of epigenetic mutations, because basically if we look at it from a perspective of a population, then if there are like changes, like random changes in the genome, then we can approximate them by Brownian process. And then if we generalize for the population, then this will give us a diffusion term. And then we have this effect of natural selection where we will include the growth of the cells, the apoptosis, and the elimination by the treatment. So let's start with this diffusion term. So first of all, we need to reflect the fact that diffusion here correlates with the number of divisions. So to include this, we are considering that the diffusion term changes over time, and basically it's proportional to how many divisions we have in the population, which is calculated by this term, and also which is also proportional to the availability of resources. So that was an interesting modification, and I think this is the first time that we introduced this term to this type of models. And the second term, which is natural selection, so it consists of three terms. So the first one is the proliferation of cells, and which depend on the availability of growth factors. So we have growth factors, and also it depends on the phenotype of each cell, which is included through this function, which depend on the genome. And then we have the elimination of cells by apoptosis. And finally, we have the eradication of cells by the treatment. So here the treatment is given by this function IT. So how do we introduce the fitness landscape? So for the multi-scale model, we have introduce it in more details by considering that the activation rate of RAS to depend on the genotype space. But here, since it's a continuous model, we do not have like a specific model for intracellular regulation, but we know that the RAS activation rate reflects on the proliferation rate. So basically, we just consider that the proliferation rate depends on the genotype of cells. And also, we consider the same branching pattern because we wanted to compare the results with those obtained using the multi-scale model. And one important aspect of the model is growth factors. So growth factors are very important because when there is like scarcity of resources, then that promotes the competition between clones. So we have a specific equation that describes the influx of growth factors, their consumption, and their degradation. Finally, we have a simple pharmacokinetics model for the treatment, which basically describes the concentration of the drugs. So for this work, we did not have like a very detailed model for pharmacokinetics, but basically we assumed that all drugs have the same effects, which is inducing the apoptosis of multiple myeloma cells, and we assumed that their aggregate effect is modeled by this concentration. And basically, we consider a protocol which consists of giving one dose for 90 days. So one dose of these 3 drugs for 90 days. So it's like a long cycle of treatments. And before introducing the treatment, we wanted to compare the obtained results with the ones of the multi-scale model. And by calibrating some parameters like the mutation rates and the growth factor availability, we obtained this graph, which is, as you can see, the dynamics are very close to the ones reported by the multi-scale model, but there are still a few differences. For example, in the multi-scale model, the eradication of less aggressive clones is not complete. So even after these clones stop being dominant, they are still there in the population. So there are still a few cells belonging to these clones in the population. And this is, in my opinion, due to the stochastic noises. So in this model, we have this stochastic perturbation that promotes the survival of the cells. And so we have a few cells, for example, here belonging to clone number 3 that remain until the end of the simulation. Another difference is that in this model, toward the end, the clone number 4 seems to be taking the lead over clone number 2, but I think this is just because of the choice of parameters. But basically, the two models. Basically, they provide the same dynamics. So now the advantage is that we have a model with a low computational cost. So it takes a few seconds to make simulations with this model, and we wanted to take advantage of that. So first of all, we wanted to see the effects of the mutation rates. So the mutation rate describes how many mutations a cell can have after each division. So what happened is that when we increase this rate of mutation, then the diversity of the tumor also increases, which means that we can have different clones in the same tumor. And also the final size of the tumor also decreases slightly, but not very much. So that was the first result that we noticed. And the second one was related to growth factors. So what happens when we increase the influx of growth factors? So, so first of all, when there is not so many growth factors, then the evolution of the tumor becomes slow. So, by evolution, I mean like the timing when there is the more aggressive clones start to appear is delayed. However, when the resources become abundant, then the effect of growth factors on tumor evolution becomes insignificant. And this is the case, for example, in these two figures where we increased the growth factor significantly. But if you can see the evolutionary dynamics, they do not change. It's just the size of the tumor which change. So basically, the effect of growth factors is more important when there is scarcity of growth factors. But when there is abundance of growth factors, then they do not influence the evolution of the tumor. And then we introduced the treatments, and basically when we introduced the treatment, we also changed the mutation rates. So what happened is if the mutation rate is low, then we have only cells belonging to the less aggressive clone, which is the clone C1, and these cells, all of them, they get eradicated by the treatment. However, when we increase the mutation rate, then there are other clones that start emerging. So when the mutation rate is very high, then there are many clones, and this leads to relapse. So there are more clones, and there is like some clones that will resist the treatment, and these clones will lead to the relapse, and there is new cells that are being that survive the treatment and start proliferating again. However, there is something which is very important, which is that if we set the mutation rate to a value like an intermediate value, then basically when we finish the treatments, we will have a relapse. However, the new tumor will consist only of clone 2, which means that there is no clone 3 and no clone 4. And the reason for that is that the treatment eliminated all the cells belonging to clone 3. So only the clone 2 survived, and these cells basically will cause a relapse and will lead to the development of a new tumor, which consists only of clone 2. So this result is very important, and it shows that the timing of the treatment with respect to the evolution of the tumor can completely change the trajectory of the evolution of the tumor. So in this case, we don't have the apparition of clones 3 and 4. So in conclusion, this model basically approximates the results obtained with the more detailed multi-scale model, and furthermore, it provided some interesting insights. In particular, we showed that the increased mutation frequency promotes the diversity within the tumor, which increases the chances of relapse. And also, one important result is that when there is an abundance of growth factors, then the effect of growth factors becomes limited. And finally, the trajectory of the evolution can be changed depending on the timing of the treatments with respect to the stage of the evolution of the tumor. Okay, so now I will move to another study that we have developed to study drug resistance, but not due to intraclonal heterogeneity, but due to some shifts that are induced by a treatment. So there are some treatments that influence the metabolic pathways of cells, which cause like drug resistance. And these basically, in this case, there are other treatments which are prescribed to reverse the effects of the first treatment. So the approach that we use here is that we have a pharmacokinetics model of these two treatments. So the first one eliminates the tumor. However, it also induces this activation of the pentose phosphate pathway. So I will explain this in the next slide. And the second one downregulates this pathway, so this pentose phosphate pathway. However, this second drug is highly toxic and has a high clearance rate. So we use a pharmacokinetics model to describe this, and then we introduce this pharmacokinetics model in a multi-scale model of multiple myeloma development, and we see the development of the tumor. So basically, the drug resistance here is induced by metabolic shifts that are induced by giftinib. So giftinib is basically an EGFR inhibitor, so epidermal growth factor receptor inhibitor. So it binds to the epidermal growth factor receptors and stops the RAK signaling. And to reverse the effects on the pentose phosphate pathway, there is this other treatment, 6EN, which inhibits this pathway, PPP, and makes the cell sensitive again to the treatments, to the first treatment, which is the EGFR inhibitor. So this type of this strategy of treatment only works when the cell does not have any mutations in the RAS or RAF proteins. The challenge is that this PPP inhibitor 6EN is both highly toxic and has a high clearance rate. So this is why we wanted to do optimization, because basically this drug, you cannot use it you cannot prescribe a high dose because it's highly toxic. And also when you prescribe a dose, then it clears very fast. So basically, we use this model for intracellular regulation that consists of the RAS-ARK cascade and also the pentose phosphate pathway. And basically, this is where the 6EN inhibit this pathway, and also the effects of the gefitinib, which is the EGFR inhibitor. So we consider that it inhibits the binding of the epidermal growth factor to its receptor. So there is this sort of competition, and now we have also another project with a more fine-grained description of this process, like this competition. But basically, this is the intracellular regulation pathways that we are considering, and they are reflected by this equation. So this one for ERK, this one for the transcription factors, and this one for the NIDPH, so which is the results of the PPP pathway, which promotes the conversion of, or the generation of the DNA. And DNA is represented by this equation. And finally, the treatment 6EN is represented by this equation, which describes the concentration inside of the cell of these treatments. So as you can see, this treatment inhibits the NIDPH in this equation. So also one important feature of this model is that we describe the concentration of the treatments in the extracellular matrix. So like this epidermal growth factor inhibitor, this gefitinib, we describe its concentration in the extracellular matrix to a specific equation. And also we use some pharmacokinetics model to describe the concentration of these two drugs, so the gefitinib and 6AN. And we consider different treatment protocols. So these treatment protocols were considered because, first of all, they are used in clinical practice, and second, such that they do not exceed the maximal dose that cause toxicity. So, and this is especially the case for this drug 6EN, which is highly toxic. So we have all of these different protocols, and we run simulations where we combine the different protocols and see what happens. So we can observe basically three types of results. So first of all, when the concentration of the treatments is low, then the tumor will keep growing. Probably the growth rate will decrease, but nevertheless, the tumor will continue to grow. In the second case, the tumor will be eliminated. So this is like the favorable outcome. And the final outcome is to have a relapse, which means that the tumor will start basically shrinking, but then we will have a few cells like this that will survive, and these cells will lead to the relapse of the tumor. So when we look at the results, so this is what happened when we combine these different treatment protocols. So these are the gefitinib regimens, and in the columns we have the 6EN treatments. So there are two successful strategies, which are here shown with this blue color. So the first one is when we administer, so there is a range of 6EN daily doses that we can administer to have complete elimination of the tumor. And second, it is when we combine this high concentration of 6EN plus high concentration of gefitinib. So it's like very intensive and high dose treatments that combine the the two drugs can also lead to the complete elimination of the tumor. Another thing is what happens when we increase the effect of drug resistance. So this is the effect where the metabolic shifts increase due to the EGFR treatment. So in this case, this therapeutic window that leads to the elimination also shrink. So if we increase the effects of drug resistance, then probably we will have less choice to make. So probably we will have only like this 0.6 that becomes effective. Okay, so in conclusion, so the model reveals that there are two strategies that work. So the first one is that there is this interval for 6 AN daily dosing, which maximize the response of the tumor to gefitinib. And the second one is combining high-dose pulsative gefitinib treatments with high-dose short-term 6 EN protocol. And basically here we show that these two hypotheses can probably be taken into experimental and preclinical studies. Okay, I think I'm running out of time, but I will quickly talk about this very important work. I think this was the first work that we did, which studied the impact of multiple myeloma on erythropoiesis. So erythropoiesis is the process by which red blood cells are produced in the bone marrow. And basically in this study, we tried to quantify the effect of multiple myeloma infiltration. So what happened when this multiple myeloma infiltrates the bone marrow on not only the function of erythropoiesis, but also on anemia. And this work, we did it with a team of Professor Mark Curry, who did the experiments, and we were able to validate the model. So regarding the model, so it builds on a previous model of erythropoiesis, so red blood cell production in the bone marrow, which basically provide this feedback that leads to the development of the erythroblastic islet. So if we look at in the bone marrow, so there is what we call the erythroblastic island, which is a structure of cells. So there is this macrophage in the middle, and then we have this colony-forming unit, erythroblasts, and we have the reticulocytes. So these are two stages of red blood cell maturation, and there is a feedback because these reticulocytes, they secrete some proteins which lead to the apoptosis of these other cells. And so the population of these cells remain approximately constant. So this is an interesting model because these cells can either have the possibility of differentiating, which means becoming reticulocytes, dividing and becoming reticulocytes, or they can also proliferate, which means they can divide and give rise to two daughter cells of the same type. So this is what happened when we made these simulations. Basically, we start by seeing what happened when the myeloma cells infiltrate the bone marrow. So they start proliferating, and also they generate some apoptosis inducing cytokines, which destroy these different cells. And after that, we introduce the treatment. So when we introduce the treatment, then these cells get eliminated. And basically, there is this colonies that appear again. So there is these structures that appear again and basically lead to the recovery of anemia. And this is exactly what was observed in the experiment. So we were able to compare with clinical data collected by our colleagues using by simulating the concentration of multiple myeloma protein and immunoglobulins. So these are some proteins secreted by multiple myeloma cells, and we used a model to describe their concentration, and we compared with this experiment. So the results were very approximate. So in conclusion, we have developed a discrete continuous model to describe the impact of multiple myeloma infiltration on erythropoiesis, and we took into consideration the mechanical and biochemical effects of multiple myeloma cells, and the results of the model agrees with the experiments, and the important results that was also observed in the experiments, but also in the simulations, is that when there is like a high when the treatment starts lately, so when there is a high number of multiple myeloma cells, then the chemotherapy does not eliminate all of the cells, and there is a relapse, which leads to the persistence of anemia. So like there is always this destruction of erythroblastic islands, and anemia basically persists. Okay, so I will finish with some conclusions, and I think one important thing to underscore, which is that this topic is very important because intraclonal heterogeneity is what causes drug resistance in multiple myeloma. And so now, unfortunately, multiple myeloma is considered to be an incurable disease, although there are some very few cases where there was a complete recovery. But it remains like there is very low chance of recovery. And so I think this is a very important topic to study, to try to understand the implications of intraclonal heterogeneity and the interaction between the evolutionary dynamics and the treatments. Here we have used these two approaches. So the multi-scale modeling provides an accurate description of the intracellular regulation pathways and so on, while the phenotype structured model is a low computational tool that can use to possibly explore a wide range of parameters and generate testable hypotheses. But nevertheless, I would like to stress on the importance of the collaboration with biologists and clinical doctors, because I think this is what's important, is to listen to the biologists and see what's their view of this topic, and what are the questions that we can study with these tools. And as future perspectives, I think one important possibility would be to include immune cells and immunotherapy into phenotype structured models. And this will allow us to also study the effects of cross immunity. So when we have like T cells and the fact that they have some respective affinity with the immune cells, and then we can introduce the checkpoints and so on. So I think that could lead to some interesting dynamics that I think we can explore. However, in general, one thing about multi-scale model is that they tend to focus on higher level physiology. So these are like cell-cell interactions and cell microenvironment interactions. However, in cancer, the disease is primarily caused by genetic changes. So I think it's very important to have more fine-grained models of intracellular regulation. And I think this is something that now we have the tools to explore, which is trying to integrate more fine-grained models of intracellular regulation that introduce the stochastic and spatial processes and see how they interact with mutations, and then try to integrate them into population multicellular models. And one way to do it is through machine learning. Okay, I think that's it for me. Thank you for your attention.
Thank you very much, Anas, for this interesting presentation. So we have some time for questions, discussion. Please, if there are questions from the audience, you can ask. Waiting for other questions, I will begin with some comments and questions. I think that really you raised many. Okay. Okay. Okay, I make a short break. Anastasia, please ask your questions, then I will continue. Anastasia, you can ask your question.
One moment, do you hear me?
Yes, yes.
It's better now, I think. My question is, does the treatment which you include in the second and third model influence equally the clones of myeloma cells?
What was the last sentence? Can you repeat it, please? Include?
Does the treatment which you include in the model equally influence different clones?
Oh, yeah, that's a very good question. And actually, yeah, this is, I think, one of the limitations that we did in this work, which is that we wanted to focus on the aggressivity of clones. So we did not want to include resistance. But you are completely correct. I think this is one aspect that probably we can include in future works, because there are some studies that
So
show
for now,
that
they equally, the
Yeah.
treatment influence
Yeah.
equally different clones.
Yeah,
I think
right.
it's interesting because in your models, treatment influence equally.
Yeah, yeah. Basically, the treatment eliminates all the clones equally, and we did it on purpose because we wanted to focus on the effect of having different clones with different aggressivity. But you are right. So there are some experiments that show that the clones which are less aggressive, they tend to be more resistant. And this is something that we did not explore yet. But yeah, I think that's a very important perspective, and probably we should explore this question. Yeah, next.
Thank you.
Thank you.
Okay, thanks, Anastasia. In fact, you took one of questions of my list. So yes, so yes, you presented really several works, several approaches to study this disease, and it's, as you mentioned, it's really very dangerous disease. And when you say basically incurable, I think that you mean that chemotherapy, that using chemotherapy, it's difficult to eliminate it completely. So as far as I remember, our discussions with medical doctors, particularly with Mark Curie, so first they use chemotherapy, several courses of chemotherapy, and after that they do surgery, right, to replace the bone marrow, because they are never sure that they can completely eliminate all cancer cells. And if some of them remain and there is relapse, so in this case prognostic is very bad. So do you remember, or can you say after the surgery, is the prognostic good enough? So how is survival rate after surgery? Do you know about that?
Yeah, I didn't look for that, but I think it will depend on the stage, because at some points I think there are like also metastasis, and I know that surgery can be effective if probably the cancer is detected during the early stages. But at some points when you have like different when there are like different lesions in different areas, then I don't think it's a good option.
And also, well, you mentioned that, but I'd like to return to this question about why these cancer cells cannot be, or as usually cannot be completely eliminated by chemotherapy treatment. Is it, as you say, because of different clones, or why is that? Because there are many different types of cancer, and in most part of cancer, there are different or maybe multiple mutations. Why sometimes it is possible to eliminate, like say, all cancer cells, but not in multiple myeloma? Do you have an idea about that?
Yeah, I think that's a good question. I don't know for the other cancers, but I know that for multiple myeloma, I think these treatments, there are different protocols, and basically each of them has different mechanisms. So, for example, this proteasome inhibitor, it influences this. It inhibits, I think, the NF-kappa B protein, and the others, they, for example, they increase the effects of immunity, and there are even some unknown mechanisms. So I read in the literature that people do not completely understand how these treatments eliminates the cancer cells. So, so I think in multiple myeloma, what they try to do is basically they alternate different regimes, and basically I think also this disease there are different mutations and different samples and things like that. So I think it's a question of complexity. Like we have like many mutations, and also we have many drugs that we don't understand what they do exactly. We know that they induce apoptosis, but they can have different degree of response depending on the cells. But I think most of the doctors and most of the literature, they confirm that it's because of intraclonal heterogeneity. So, yeah.
You also mentioned immunotherapy. So do they already use immunotherapy for multiple myeloma? I just don't know about that.
Yeah, I think I think they do. I think there is some. I think it's still in clinical trials. Probably it's not yet in. It's not been applied yet for the treatments. But I know that there are like this program Det1 and program Det ligand 1, and also this other classical immunotherapy drugs that are being tested in the clinical trials, but I don't think it's been used in for the actual treatment of patients yet. Yeah.
Yeah, because we should know that, of course, immunotherapy is still considered, maybe now a little bit less than before, but it is still considered as a very perspective and very powerful tool. But it is very expensive, and it also strongly patient dependent. So, so it's not easy to use it. When you presented this competition of different clones and emergence of different clones, you told us that the mutation rate is very important. Is a very important parameter. In particular, for high mutation rates, there are more probability of the emergence of these other clones, which can be, in fact, more aggressive. So again, mutation rate is an important parameter, but what it depends on, because shall we consider mutation rate as like mutations as just a random process, just it happens, we don't know how it happens, why it happens, or it depends on something that can be controlled by something? So what do you think about that?
Yeah, that's a very good question. I think probably it's something that occurs during division. So when there is like replication of DNA, and what could influence this? So probably the signal in itself. So if there is like more DNA replication, then I think also the mutation will be increased per each division. So yeah, I think that's probably the main factor that I could think of. I also think that there are these genetic drugs that try to alternate the mutation process. I don't know how they work, but yeah, probably there are some ways of influence, because it's like in the gene expression. So it's something like genetic.
Yeah.
So probably that's also there is some genetic factors influencing this process.
Yeah, by the way, you're saying that, you make me think about other data about hereditary, like properties of multiple myeloma can be hereditated. I mean, you understand, from family connections, or there is no data about that?
Yeah, I haven't looked into this question, but I think most of what I read is about it. I think it's related to the environment. It's the risk factors. I think that's why it's like in developed countries where probably there are like more pollution, or like also like genetic modified food and things like that. So yeah, I think most likely it's related to the environment, but I have to check the question of whether it's genetic or no. I don't have like a clear answer for that. Yeah.
Well, yes, there are many other questions. It's really quite completed, quite interesting study with different models, different approaches. So my last question, not to make this discussion too long. What do you think is these different approaches of multi-scale modeling, like individual-based model, hybrid models, continuous models, their combinations? So to what extent it can be used also to other cancers? So what is specific, what in all this modeling approaches you presented, what is specific for this particular type of cancer and what can be generalized for other types of cancers?
Yeah, that's, I think, very important question, and probably it will require some reflection, but I can say that I think the particular challenge of multiple myeloma is that it's genetically complex. So there are like these different clones, and how to integrate these different clones into like a single framework. And this is something that's also the case for leukemia, for example, and lymphoma. So like this hematological cancer, they have intraclonal heterogeneity. So I think the same analysis probably can apply with some modifications related to the involved pathways and so on. Also, I think that basically this phenotype structured modeling, I think it's very powerful tool, and I think that there are like many interesting use cases probably in some other cancers. Because we know that these relapses and all of that, it's due to the emergence of new clones. So I think I'm pretty optimistic that probably it can be applied to different cases. But as I said, I think the challenge is how to integrate this fine-grained intracellular regulation into multi-scale models. So I think this is a very important question, and so also I'm working on this question with a PhD students by using machine learning. So basically we have this single cell model, which is like a 3D model for with Brownian dynamics and everything that we use to generate data, and we train a machine learning, and we integrate this neural network into like this other cells, and this allows us to run like fast simulations. Yeah.
Yeah, it's really very interesting. Also, what I was thinking, and I'd like to briefly mention, because since multiple myeloma concerns plasma cells, and we know that plasma cells are very much involved in the immune response. Mm-hmm. So I think it's also an interesting question to think about the interaction of, so there are different possible interactions between multiple myeloma and immune response, and not only immunotherapy, but also the immune response of our organism to infections. Probably it can be modified or somehow influenced by multiple myeloma. And another also question, of course, often discussed in cancer modeling in particular, is angiogenesis. And and in fact, many years ago, when we started to work on leukemia modeling, I did not really realize at times that angiogenesis concerns is involved not only in solid, what we call solid tumor, but also in hematopoietic tumors. So I don't know, we did not discuss this question, but maybe angiogenesis can also be taken, should also be taken into account in particular leukemia or multiple myeloma. Anyway, thank you very much, Anas, again for this very interesting presentation. Yes, sorry.
Thank you for the suggestions.
I would like to know that we have a question in the chat. One more question.
In the chat. Okay, because I don't see raised hands, but in the chat. Okay, let's see what is in the chat.
Oh, I can see it.
Yes. So, yeah, if you can read, yes, please.
Yeah, so you talked that you included noises to the model. I wanted to know at which level we can see it on your equation specifications. Yeah, this is a very good question. So the structure of the multi-scale model is a little bit complex. So I can think about two levels of noises. So the first one is for the cell cycle duration. So the cell cycle duration that we consider is not fixed. So there is like a perturbation that we added to make the difference between cells, which is taken from experimental data. And the second one is in this equation of the phenotype. So this equation. So after each division, there is like an addition of a perturbation, which is basically sampled from a uniform distribution. So these are the two stochastic noises that I can think of. We didn't use any stochastic differential equations or anything like that.
But also, when you consider the model of cell motion, do you have stochastic terms there in cell motion?
I don't think we have one, but I think probably when
because sometimes it is added also to the equation of motion.
Yeah, I think in this case we didn't have any stochasticity in the forces that are exchanged. But on the other hand, we had probably some stochasticity while introducing the cells. So the location of the cells probably was sampled at the sampled from uniform distribution at the beginning, which can also count as stochastic noise. Yeah.
Okay, very good. If no other questions or comments, thank you very much again. Thanks also to all listeners. Have a good day, everybody. Goodbye. Thanks, Anas.
Thank you.
Hope to see you soon again. Yeah, bye.
Thank you very much. Goodbye.
Goodbye.